Awesome. I believe we're live. Cool. Well, thanks everyone who's joining today. Looking forward to a really fun and honestly important topic in this conversation, which is basically around how we gas the engine of longevity research, but also longevity products. So joining us today, we have two pioneers in the field who are definitely doing their part to accelerate longevity research and products. So we've got Jillian from the molecule side, and then we have Krister Kauppi, who can introduce himself, as well as Mitchell Lee. So yeah, go ahead and introduce yourselves, then we can get going from there. Krister, do you want to go first?
Yeah, I can start. I'm located in Sweden, Gothenburg, and I have a long background in system and business development. And I like to view things from a system point of view, and through that lens, try to solve problems with the help of IT and the development of processes. And yeah, I'm also founder of the Rapamycin Longevity Lab and the founder of the Rapamycin Longevity Series podcast.
Awesome. Go for it, Mitchell.
Hey, everybody. My name is Dr. Mitchell Lee, CEO and co-founder of Ora Biomedical. I am a scientist turned entrepreneur, so I learned about aging research by studying it on the benchtop, identifying and testing interventions that extend organismal lifespan, and becoming kind of frustrated with the slow rate of how those fundamental discoveries were moving out and being translated out of the lab. So that led me and a group of other pioneers in aging research to form Ora Biomedical. Our goal is to facilitate finding the best breakthrough, game-changing, disruptive interventions that extend healthy lifespan and fight the diseases associated with aging all at the same time. And I'm coming from beautiful, overcast Seattle today.
Never not overcast. Cool. So I guess opening question to get started, Mitchell, why did you start Ora Biomedical?
Yeah, so I was a grad student in Matt Kaberlein's lab, who's a pioneer in aging research. He's one of the first individuals to make connections between mTOR signaling and aging, which is kind of the gold standard molecular pathway for influencing lifespan right now. And so I was in his lab thinking about what the world looked like. As I saw it, we needed to put resources and effort into finding these interventions that can target the fundamental mechanisms that drive aging. And there really wasn't a big, strong influence or engagement in doing that in academia. And, you know, that sounds like maybe, okay, whatever, do it somewhere else. Academia is the place where research science happens. It really doesn't happen in too many other places. It's largely federally funded. And if you don't have enthusiasm in academia to do research, you're not going to get the funding to do the research, and it's not going to happen. So at Ora Biomedical, we think we've lost about at least a decade of interventions discovery process with this misalignment between academic goals and what we need to do to move these interventions forward. I was seeing that, getting frustrated with it, figuring out what my pathway would be, and an opportunity arose through Matt Kaberlein's lab to take the lead at Ora Biomedical, spin out the Wormbot technology from the lab, which allows us to identify interventions quicker and at better quality than anybody else, and really run at this goal of finding the breakthrough interventions that are going to transform health in the 21st century.
Awesome. Great overview. So definitely a few things we could dig into there, but I guess maybe the place to start is like, what is the Wormbot? Why is it so transformational? And why does it kind of solve the problems that you were seeing in both academia and in industry?
Yeah. So first, to take a couple steps back, we do our fundamental discovery process in worms, or C. elegans, a small little millimeter-sized animal. If you look up online, you'll see a bunch of cool pictures and videos of them. And so we look for interventions that extend lifespan in that model. And the first question people ask is, how do you possibly learn things that can translate to humans by studying something in such a seemingly disparate animal or something so different than a worm? At the end of the day, the things that drive aging are so highly evolutionarily conserved that the same things that drive aging in a worm drive aging in a human, and a companion pet, and a mouse. So it's actually remarkable that there's this robustness in aging phenotypes that have this translation. So that's the first step. Why worms? Now, why can't we do it faster than anybody else? So in academia and in research as it is, longitudinal survival studies are done with people looking at dishes of animals and actually checking off in an Excel file whether they're alive or dead. It's a very slow process. There's been really no automation done around it. And that's where the Wormbot comes in. We have a robotic system that does automated population level imaging. So we take pictures and videos of populations of animals from their birth all the way until their death. And we do that at a scale that is exponentially larger than just about what anybody else can do. A single one of our Wormbot platforms does the work of about 5 full-time research scientists.
Wow. Incredible. So I guess, you spoke a little bit about C. elegans. Maybe one thing to do super briefly is talk about the features of it that make it such a good model organism. So in your mind, like why worms? Why not mice or flies or these other model organisms that people are used to learning about in biology class?
Yep. So thinking about that evolutionary conservation, if you looked at 2 cells and I said, this one's a human cell, this one's a C. elegans cell, and I just showed you pictures, it'd be indistinguishable. You couldn't tell the difference. They've got all the same organelles, subcellular features, and they do all the same processes. At the end of the day, a cell has to perceive nutrients, figure out how to take in those nutrients, convert them into the cellular building blocks. That generates waste, and you got to get rid of the waste. At that fundamental level, a C. elegans cell does the exact same work as a yeast cell, as a human cell. And so the kicker there is that's also where a lot of this age level dysfunction starts to arise, is in those fundamental cellular systems. So the worm gives you an opportunity to look at that, not just in cells. They also have reproductive, muscular, nervous systems, all the different systems that we think about are important for aging. They have a lot of those systems as well. So they're great small animals to make these fundamental discoveries in. The question of why not go for flies or mice or something like that, all model systems have some fundamental limitations. I've just spent some time telling you about how we're all connected to worms, and it's awesome. There are differences too, and model systems all have limitations due to those differences. So it's really where you pick and how you move forward. With us, we chose C. elegans because we can build our robotic systems and do automated tracking of these animals and get incredibly sophisticated data. And they're also incredibly cost effective as well. They're the least expensive animal to grow thousands and thousands of in a very small space to do these kind of tests.
I think also you can mention that the lifespan of a worm is quite not so big if you compare it to a mice or something like that. And that way you can also get a really nice cost-effective approach in testing interventions in worms.
Yeah, 100%. Yeah, and C. elegans lifespan's about a month. A fly is about 2 to 3 months. A mouse is about 2 to 4 years. So yeah, we wanted to do it in the quickest system that we could. So yeah, Krister, that's an excellent point.
Yeah, so we kind of call this on our side the longevity trilemma, which is like the cost, the speed, and the quality of the data that's coming out. And basically, if you can think about longevity research as a computer, you're trying to maximize the bandwidth of information through this system. And so, to your guys' points, C. elegans and the Wormbot seem to be the most efficient way to get maximum bandwidth into the system. So I think you guys put that really well. But yeah, maybe Krister, because you just jumped in, would love to hear more about your longevity story. Like what got you into this space, and tell us more about the Rapamycin Longevity Lab as well.
Yeah, I have a big passion in the longevity area, as Michelle and all the others here, and I feel a big frustration also that things are moving too slow forward. So we need really to speed things up in different ways. And one interesting thing why I started, for example, the Rapamycin Longevity Lab is because of Ora Biomedical's launch of the Wormbot technology. Because one thing that is super interesting with that technology is that you don't need to be a longevity researcher to conduct longevity studies. I think that is a super interesting part here. So, and which opens up that much more people can come into the field and help out in moving the field forward. And when it comes to the Rapamycin Longevity Lab, the focus of the lab is to take mTOR inhibitors to the next level. And this we're going to do by combining different interventions with mTOR inhibitor, because we think that one single intervention will not have the biggest longevity effect. Most likely, the biggest longevity effect will be achieved by some kind of combinational therapy. And thanks to the Wormbot technology, we can start to test combinational therapies and not just single interventions. So that is one very interesting opportunity that has arisen, and which the Rapamycin Longevity Lab has jumped on.
It's super interesting from the drug development standpoint. You spend a lot of time looking at like one compound, and you think about how long it takes to get that one compound all the way through the process. And that's if you're successful, right? Like you have to do trials at many different stages, even getting to human trials that can in totality take 10 to 15 years. And so hearing this combination and, testing more rapidly different, I don't know if cocktails is the right word, is so important, because that's really, not only are you accelerating the data, but you're actually accelerating like in an exponential way, because you can find out if things work by themselves and if there's an exponential effect, potentially if things work more strongly together to get to the ultimate unlock. But doing kind of one at a time is, I think we would all agree, is far too slow and is takes a really long time and is risky, right? Like you're putting all your eggs in one basket just to see if one thing works. So
but
I love that.
Yeah, but I think also it's important to do the one interventions to see what effect can one intervention have, and then we start to combine also the intervention with each other to see, can we get the synergistic effect, additive effect, or even in some cases, which we have seen, detrimental effects. Because even if two really good interventions work by as a single interventions really good, but when you combine them, it can be that the dose of like mTOR inhibition gets too high, and by that you get the detrimental effects. So yeah, it's a super interesting area, this.
So how do you find the space? Oh, real quick, Benji, how do you find the space like receptive to this paradigm shift? Because I think it really is, science and how we do science is deeply entrenched. So how do you think about, talking to others in the space and really coming with something like Wormbot and this more high throughput science? Do you find people really open to this? People are eager to test this out, or is it kind of like we want to let that play out first and see how it plays out in the long run before we, really get on board with that as like a process or a procedure?
Yeah, I jump in on that. So yeah, the first part is just like what Krister was saying. So with the Wormbot, it allows us to do science in a way that's really disruptive. We actually bring science to the people, and anybody can be a researcher. If you go to the Ora Biomedical website, you can look at the Million Molecule Challenge, and you can be the scientist. You can propose an idea and have an intervention tested. Not just a single, a combination. There's so much I could say about that. But in terms of the enthusiasm and interest, that is really where we're seeing this incredibly exciting signal. People want to participate in science. They want to have this process demystified. They are not excited with the slow rate of progress that we're seeing. And there is an opportunity with our system to actually take the lead, accelerate, grab the reins of science. Let us be your scientific team, and we'll test your ideas. So that has been so powerful. And what we want to do is just spread the word on that, get more people to get engaged and really understand the power that they have. As a quick aside, the interventions that have been sponsored by just citizen science and the Million Molecule Challenge, I never would have thought to test. Nobody else on my team would have thought to test. People are bringing ideas and insights to the table that we would not have prioritized. So your ideas are important, critical, and this is a place for you to vet those hypotheses. In terms of the powers that be and the power structures and biopharma development and things like that, we're still figuring out what that playbook looks like. And biopharma is the most entrenched of these kind of systems. They're going to be the hardest to change with time. Having said that, there is movement happening in that biopharma space around longevity intervention. So there's more and more excitement around it. People are figuring out how it aligns with this current biopharma model. And I think there's important ways that longevity technology aligns. I think there's important ways that they don't align, that we have to figure out other solutions around.
Maybe that's a great jumping off point, honestly. Because maybe these pharmas are super excited about longevity interventions. I know both of you are. I know there's a lot of people probably listening to this that are waiting for more longevity products. They're not necessarily supplements, but they can't really be considered drugs either because they don't necessarily treat a disease, because aging isn't considered a disease. So, all of this is to set up a question, which I would love to hear from both of you, which is, if you could design a regulatory system for longevity, how would you do it?
Krister, I'd love to hear your thoughts on that first.
I think one important thing is to get the infrastructure in place, and the thing that Ora Biomedical has done here has taken the first step forward in open up technology for the public. That is a super great step forward. The thing that I will really like to see is that we start to automate much more things around preclinical studies. Like we should have, for example, a yeast bot. We should have a fly bot. We should have a mice bot and things like that, so that we can conduct these longevity studies in a high efficient and cost-effective way. And you should not need to be a longevity researcher to use these services. So I think that is like the base thing. And then if we come to the human clinical trials, here we also need to do lots of improvements, because it's very cost heavy to do a clinical trial from phase 1 to phase 3. And there are interesting development in this area. For example, the network states has arisen as a reaction to this, where, for example, pilot trials can be done in a cost-effective way, and by that we can also test things so that we can do the phase 1 trials in a much better way, and also minimize the risk of phase 1 and phase 2 trials. So I think, we need to get the pipeline as good as possible and work from there.
Yeah, the place I'd jump in on it at is, the pathways and the regulatory constraints as they are a challenge, but I don't see them as insurmountable. The biggest challenge is figuring out what you measure. What are the biomarkers? What do you do to say that I've done this intervention and now I'm better off than I was when I had? How do you start understanding that? Once we start getting those kind of biomarkers in place that are highly predictive of biological aging, then I think a lot of these structures are going to start to understand how to work with it. Right now the situation is so interesting because longevity biotech applied to biopharma is really just saying we've got a new kind of target that we're going to aim at disease. There are targets that are associated with hallmarks of aging. Hallmarks of aging have some connection to aging. That's a great argument to have among scientists with a beer. But then what you ask is, from that, you've got a new target, you're aiming it at disease state. So the disease is the end model, the patient is the beneficiary. That's amazing. With geroscience, there is this connection between aging and the diseases that ultimately limit our lifespan and even other diseases. The issue with that, so that is going to result in new breakthrough therapeutics, I have no doubt. The problem is it's hard to make that leap to a world beyond patients, where everybody's the beneficiary of longevity technologies. So the biopharma model doesn't have the playbook to make that translation. On the other side of the fence, you see dietary supplements and things like that being able to go straight to the market. And they actually are interested in aging and longitudinal studies. It's not going after a disease state and finding things that impact an indication. It's actually aiming for what we're most excited about, aging and targeting aging. The problem there is that it's a big mixed bag in terms of the quality of the research that's done. So you have to hold yourself to the highest quality standards of your science and not get over enthusiastic about taking your asset to market. Really making sure that you're doing the good homework and showing that it's impacting aging. So we're in a weird place, but it's kind of weirdly hopeful in the two different major arms.
It's interesting. We have some comments streaming in, and I just want to take a moment to read one because, Mitchell, I think exactly what you said is what we're hearing from the community. Someone said, we need to be able to understand multiple levels of organization as well as the factors that influence each and the degree to which these factors can influence one another. So to your point, like understanding biopharma, biotech, understanding, the FDA regulated path, the supplement path, and going back to even the conversation on cocktails, like, how do things work by themselves? How do they work when put together? So yeah, just wanted to take a moment to pull this up because it's a very relevant sentiment that people have in the space for sure.
It's huge. And another major thing that doesn't have the attention to it that it deserves is natural genetic variation. Even when we find these interventions, just like aspirin doesn't work across the board the same way for everybody, these interventions are not likely to be magic bullets that equally work as well for every single person. So we really actually have to develop a toolkit of these interventions along with precision approaches to apply those interventions to the right people who will maximally benefit. So it's not just like, hey, this study came out, it says X is great, I'm going to run to the store and buy a whole bottle of it. That goes into my conservatism in the space, but that's a different question.
And I think also it's not only just that the interventions can differ between people, it can also differ between situations. If you, for example, have gone through a surgery or something like that, then, for example, taking a mTOR inhibitor directly related to that is probably not the best thing to do. So there are lots of things to get in place, and Yeah.
Yeah. So honestly, really love where this conversation's going. I think, your guys' answers to the previous question was super interesting in that what you named is potentially a bunch of endpoints that we could be testing, and I think what we all agree upon is that worm movement and lifespan is that first endpoint we want to test. But there are tons of other endpoints we could do, like fly movement and lifespan, mouse movement and lifespan. But then when it comes to the humans, all of a sudden everything gets very murky, right? There's a bunch of different potential options we could have. Everyone has their opinions. Usually it's based on whatever company or research they're working on. But frankly, this is where we get super excited about creating a prediction market out of this whole thing, because the reality is we don't know. We need to find out as a species, one, if we're going to get this piece right, but also two, if we're going to speed up the rate at which we can do longevity trials, right? Because we need essentially, like in the markets, what are called leading indicators, right? Before human death, how can we start to measure things, that are nearer term, that allow us to get signals faster and allow us to iterate faster and kill, cocktails sooner and advance, better ones sooner. So this is honestly one of the big impetuses for starting a prediction market around this whole thing, is because you basically, scientists are all very skeptical. They're all extremely biased, and we want to create like a neutral playing field and an arena where we can test these things out in the open, and just let people put their money where their mouth is. So that's kind of the reason for creating the prediction markets, is because these questions, the answers to these questions are so important, and people do have a vested interest in them, but they're unanswered.
Yeah, that's another power of the Wormbot system. Right now, we look at live and dead, whether the animals are still around or not. We also look at about 20 different health, behavior, movement phenotypes that change with age. And our system is powerful enough to be able to look at all those across the entire lifespan of the animal. So we're getting these phenotype, functional level measures of aging and how drugs change all of those features. So it actually is the right kind of laboratory to find those leading indicators and start to get into those biomarkers.
Totally. I guess I have a question around those human biomarkers, though. So from both of you having studied the longevity field and all of the aging biomarkers, methylation, functional measures like VO2 max and things like that, if you were betting men, what would you bet on is the most important leading indicator of human longevity for testing one of these new cocktails?
Loaded question.
I think this one is super hard to answer, because one of the biggest challenge that we have in the longevity field is that we don't have any good way to validate in humans if an intervention works or not. We have different clocks and things like that, but they're not perfect. We have different biomarkers, they're not perfect. So the field is trying really hard to find that measurement which we can use to, in an efficient way, validate if intervention works or not. When we find this measurement, then I think the field will really take off. So that is a very important thing to solve when it comes to human clinical trials, I will say. But yeah, that's not the answer to the question, but yeah, over to you, Michelle.
That's a hard one. Yeah, absolutely. What do you look at? So we are very compelled by functional phenotypic readouts. If aging has been modified in a certain kind of way or even reversed, it should manifest in the day-to-day lifestyle of the animal in question. At least that's what I think about when I think about a successful intervention. Like if an intervention I take changes methylation profiles and it doesn't change how I get out of bed or run around or something like that, I'm not very interested. So whatever it is, I think it needs to be tied to a functional phenotypic kind of measure, maybe something like VO2 max, some of these other things that are very easily measurable, especially with some of the wearable technologies. If we had that right connection there between easy to measure and related to biological aging, that would be such a huge breakthrough. If you ask me to throw in my bias, I'd say we should look at vascular permeability. I think probably the cardiovascular system is one of the rate limiting organ systems for humans. So looking at some of those basic measures is where I would put my bet at. And that foreshadows a little bit of where Ora's putting its bets at too.
Super interesting. So just briefly on that, we don't have to go too deep on that. I'm sure it's a whole rabbit hole in and of itself, but how would you hypothetically measure that without going and doing a biopsy and, doing pathology?
Yeah. So this is actually leveraging some of the advances that are happening in tissue modeling technology. Throughout the 20th century, we were really good at growing essentially fibroblasts or cancer-like cells in a dish. And we made some fundamental insights because of that, but there's big limits, like especially if you're trying to think about organismal complex aging and modeling it in disassociated cells in a dish. It's not really clear what you're going to get. The new models that are starting to be generated actually allow you to create small little micro organ systems or organoids. Some of them are like small little balls of neural cells that have their own firing and interactions with each other. The stuff we're really excited about is growing vasculature in microfluidic dishes. And this is where the technology is most advanced. Funnily enough, is building vasculature. We've gotten really good at doing that as scientists over the last 15 years. We haven't translated that into measuring aging and modeling aging in donors in that kind of way. And I think that's where we're going to be aiming some of our systems at in the future. It's definitely TBD, under construction, but that's where we're aiming for.
Awesome. I mean, from a data perspective, makes perfect sense. Cardiovascular disease is the #1 killer. So, looking at the cardiovascular system makes a lot of logical sense.
But I came around to this looking at some of the research that happens in the context of Alzheimer's disease. So we have been trying to fight Alzheimer's disease by targeting A beta for decades. And that's like trying to fight a fire by targeting the smoke. The fire is a metabolic breakdown that happens decades before. And the site at which that seems to kick off is at this neurovascular unit. Where this breakdown starts to arise first is in that interaction. So increasing vascular health, maintaining that with time, might end up allowing you to preserve that neurovascular interaction longer, hopefully staving off or completely mitigating these kind of neurodegenerative diseases.
Awesome. Yeah, I mean, we've actually got a project listed on our crowdfunding platform Catalyst called Project Accelerate that's doing proto-tissues. So it's basically somewhere in between a biogel and an organoid. But one of the milestones there is getting vasculature in neurons and basically all the supporting cell types as well. They're basically trying to reconstruct the anterior cingulate cortex ex vivo and then be able to ideally transplant that into a human rather than treat the existing diseased tissue. So it's more like the replacement therapy. But anyway, that is a whole can of worms.
Awesome. Yeah. We're on the cusp of so many cool things.
I was going to say one thing that really has kind of resonated with me in this conversation is the importance of a metric that people can kind of rally behind. In the crypto space, one thing that to me, I know it's more relevant to our side of the camp, if you will, but as crypto technology and blockchain technology was being built, people were having a really hard time figuring out like which projects were the most successful, which technology was the most successful, which ecosystem was the most successful, until there was a metric that everyone kind of uniformly adopted, which was TVL, total value locked. And it measured the amount of dollars locked in a protocol and kind of standardized that metric. And then once there was that metric, right, then everyone used that to kind of say, we're all working from the same yardstick. Which one's the best? Which one's, has the most users, the most adoptions, most liquidity. And to me, it sounds like a really interesting parallel in the longevity space, because it's like right now that story, there's so many different micro narratives and everyone's having a hard time like rallying behind one number or one thing to then create this like, kind of groundswell momentum behind. So like I said, more interesting, not interesting, but more relevant than you can imagine in the crypto space, because we kind of had the same like cold start or like, inertia that we were all working against until there was this clarity on like a metric. And then all of a sudden, I mean, there were like dashboards built that people were just looking at, like all the protocols that were being built stacked against this one metric measured against TVL. So yeah, I just, to me, there's a huge opportunity to create like one easily digestible metric. Not that that's the only metric or the only metric that will matter, but to help create momentum for this story that we're all, ruthlessly trying to solve.
I think one basic metric when it comes to other non-human studies is the lifespan studies, because there you have a quite simple metrics if the intervention works or not. But that is very hard to apply to human studies. But yeah, do you have any thoughts on that, Mitch?
Only just to say that, yeah, that I do think that is the important thing that will create this groundswell. In terms of what we look at, I don't know yet.
It's super hard, right? Because it's extremely context dependent, and no one wants to say that or hear that because people like simplicity, right? Of course, we'd all love if there were just one metric to rule them all. But the reality is, VO2 max is the best correlate with lifespan. That being said, if you had a drug that could fend off cancer, you probably wouldn't be able to see it in VO2 max, but it still might increase longevity. So it's still not a perfect indicator. But anyway, the truth is that the jury's out, and I think it's on us, and ideally this system that we hope to participate in and create to find answers to these questions.
Yeah.
But
there are lots of people working on this, so I feel confident that they will find a solution to this in the quite near future. Even if it's not the perfect measurement, I think they will find a good one which can unify the field in some way.
Yeah.
So.
Yeah. I would say an unmet need that, when people start talking about this stuff, one of the things that like they jokingly suggest is like, well, we can't just track people through time on these interventions. We can't do a big longitudinal study. It'd take decades. Like, well, if we would have decided to do that in the 90s, we'd have some data by now. Like, kind of like the reason why we understand that the cardiovascular system does look like it is this rate limiting system in humans is part of the Framingham heart study. It was this decades-long study that's on its like 70th year now. It's the reason why having a heart attack isn't a death sentence now. So like, it's like, really at what point in time do we want to get serious and do the hard thing? And then the question then is like, okay, if you've signed up to do the hard thing, what's the intervention? What are you going to give to people? What do you have the confidence around? And yeah, that's another kind of story. But, it might just be the hard work of actually monitoring this for a lot of people for a little while.
Totally. I guess, so one thing that we did gloss over quite a bit, but we did touch on, but both of you guys are experts on, and I definitely want, to give you the opportunity to like flex your knowledge on, is the mTOR pathway, right? So you kind of alluded to it saying that, this is one of the highly conserved pathways in biology across C. elegans, worms, all mammals and vertebrates. Every one of these species seems to have mTOR. Why did mTOR evolve? What are the known drugs that target it, and why is it relevant to longevity?
Cool. Oh, this is something that I find just so endlessly fascinating, because the mTOR pathway is so cool. It's one of these master regulators of nutrient signaling. So we talk about it as an independent axis, but it really interacts with other things like AMPK signaling, insulin signaling, all this stuff associated with just understanding your nutritional state. And if you think about it, you need to understand your nutritional state to understand what your decisions are. As a cell, I make the decision of dividing or not dividing. If I'm got the energy and the nutrients and the signaling is telling me the time is right for that, then I can divide. That's important for development. It's not always important. Sometimes that kicks off cancer if it's too much or out of whack. But the other cool thing happens when a cell perceives a low nutrient state. It's called hormesis. It's this idea that the cell says, no, it's not a good time to divide. You should instead play a defense game. You should upregulate things associated with protecting your DNA. You should turn down protein translation because that generates a bunch of waste. You should get rid of the waste that you've generated. So upregulate things like autophagy and lysosomal recycling. So this one thing of changing your perception to nutrients has this profound metabolic rewiring that happens all the way down at the cellular level. And it seems like mTOR signaling is the best rheostat. And it's something that we can actually hit and modify. So that's the coolness of the biology, the interestingness of the drugs and stuff. And I'm going to give Krister a lot of space to talk about this. But we've got rapamycin, and this is this drug that extends lifespan across all these organisms. We only know about rapamycin not because it's the best mTOR inhibitor out there, and there are hundreds of them, but just because of the inertia in academia, nobody could get funding to look at the other ones to see what's better. And so, Krister, I turn that over to you after that. I'm curious to...
Yeah, but it's as Mitch told, the mTOR pathway is a really interesting pathway because it works in different species. We have also some interesting data in humans, but there is a big gap in the field. We don't know if there is better mTOR inhibitors than rapamycin because no one has tested this, which is very strange because currently rapamycin, the compound, is the most promising longevity compound that we have. Why have we not tested if there are better mTOR inhibitors than rapamycin? And that is also one reason why I started the Rapamycin Longevity Lab, to see if we can find better mTOR inhibitors than rapamycin. And one project that we have started fundraising for is to screen 568 mTOR inhibitors to see if we can find something better than rapamycin, and also fill this big gap in the field which we have. One other interesting thing is that when Ora Biomedical launched the Wormbot technology last year, the first thing I did was to take I think it was like 5 mTOR inhibitors that I found in their library, tested them to see how well they worked. And the thing was that thanks to that, we found one mTOR inhibitor which is better than rapamycin. So yeah, step by step we move forward. So I think, or I'm confident that we will find even better ones when we screen the 568 mTOR inhibitors. So yeah,
amazing. Yeah, we're confident too, and the data speaks for itself. I think you got like second place on the million molecules challenge. So the proof is in the pudding. But
yeah, I think I have the first place now with combinational therapy with the mTOR inhibitor and glucose regulator. So yeah,
let's keep it going. Let's keep pushing that upper limit. But yeah, I think one of the questions you asked is like, why isn't there more research into these other derivatives of rapamycin, these rapalogs, these other mTOR inhibitors? And I mean, if you look into where the research funding has gone into these other mTOR inhibitors, it's all been directed at cancer for the most part, right? And it just seems like mTOR inhibition doesn't work in cancer. Okay, but still, there's this really powerful, mechanism of action that's useful for longevity that might not be applicable for oncology. And so, yeah, the existing system, as we've kind of alluded to, doesn't really have the infrastructure set up to enable funding for anything other than a disease. So it may very well be that all mTOR inhibition does is, do immunosuppression in the case of organ transplants and extend life. But still, it deserves the attention for the latter and the research funding. And so hopefully, we can help, create kind of an engine there as well.
I think one other thing that can be good to point out that if we, for example, look at what dose protocols are used, for example, in cancer treatment and organ transplant and treatment and things like that, those doses are quite high doses compared to doses that have beneficial effects. I usually compare this to exercise. Let's say, for example, that you, Benji, go out and run a marathon every day. Okay, for a short term, that will probably work. But after a while, that will have detrimental effect. It will have detrimental effect on your muscle. It will have detrimental effect on your immune system. Yeah, it will have lots of bad effect. But if you can find the right dose of running a marathon, then you can get the beneficial effect instead of the detrimental effect. The same thing is with the longevity interventions. It's about finding the right dose protocol.
Totally makes sense. So you can absolutely overdo mTOR inhibition. Too much mTOR inhibition isn't necessarily good. You want to strike that Goldilocks zone that optimizes for autophagy, doesn't necessarily inhibit the immune system all too much. But anyway, I think we have several questions in the chat from listeners. Do we want to get to some of those? I saw there were a few.
Yeah. So I think this is going back to, Mitchell, a comment you made in the beginning, like making science open, and the fact that you don't have to be a longevity researcher to advance research in longevity. We had a question from somebody that said, how can other scientists, clinicians get involved in this space and even in the DeSci space? I know you guys are kind of making a move over into opening up science with us, but just would be curious to hear from you guys, like how other people can get involved, knowing that you don't have to be a part of like a big organization or, a specialist in this space to advance longevity research. Great question. So one side is anybody can be a scientist with us. So we've totally broken down all the barriers to doing world-class research, and there's a team ready to work with you. You got the idea, we got the time. The other side is how a scientist, a clinician can start moving into these spaces. We desperately need outreach. We need people who understand a lot of this stuff at this nuts and bolts level to go and talk to people, tell people that this isn't a matter of science fiction, that it is ongoing. And those people also have to have the highest integrity and internal standards for what they move forward. So, we can't just be a lot of scientists in the space spouting their biases. Like it has to be groups who are absolutely committed to high integrity scientific communication and outreach. So I think that's where the biggest benefit can happen, is just spreading the word in a way that doesn't just, doesn't get over subsumed by the hype and everything. Because like the good news is great. The good news is we're going to transform human health in the 21st century. There's a medical revolution that's going to occur in our lifetimes. Like that's incredible. But like, the hype side of it is today we're going to make you immortal, or we're going to decouple you from, mortality and things like that. That's too far. We don't have science behind that yet. But, the good news is great. And like, we need to get people to understand the good news.
And I think also it's important that we start to collaborate in different ways, because there are lots of different initiatives out there. Let's say that you are a physician. There is a great network for physicians, which is called Longevity Docs, where they can connect with other physicians who want to enter the field. If we look, for example, at investors, they can join, for example, and connect with you guys at the Molecule, because that way you can help them to invest in research projects. Then we have, for example, the tech people. There are lots of things tech people can do in the longevity field to improve, automate, use AI and other things. So we really need to get different disciplines into the field so that we move things forward in a much better way, because this field is, as Mitch just told, it's not only for longevity researchers. We need other disciplines also.
Definitely. We had a couple questions too, to kind of like piggyback off of that idea, Krister, around like how long it would take for, you know, some of this stuff to play all the way out. I know we touched on it in the beginning, but what I do think could be interesting, Krister, from your standpoint, like you mentioned that you had, I think it was 5 mTOR inhibitors that you tested, and what that, or was it 500?
No, and the first one that I tested was 5, but the ones I'm going to test now is 568 mTOR.
What would that process have looked like if something like Wormbot didn't exist? Like, do you have any kind of speaking to the flip side of that coin? Like, we're talking about this amazing technology, but I do think for people who aren't as familiar with the process and how long this stuff can take, like, what would that look like if this didn't exist or hadn't been developed?
I think it will have been very complicated thing. The first thing you need to find is some kind of researchers who can conduct this type of, it's quite a huge screening project, and the cost of that will be quite much. Mitch, you probably know how much it will cost if you do it manually, but it's my guess is like it's 10 times more than what it costs to test it in the Wormbot technology. Because if we look, how much does it cost to test one intervention in the Wormbot? It's $100. $100. That's nothing. So you cannot get something tested by any researcher for that price.
Yeah, in terms of time frame, you know, a given grad student doing a longevity study on the benchtop would be measuring about 10 different interventions or treatment in tandem in their populations. It would take them about 3 to 4 hours a day to go through and manually look at every animal and assess life and death in those populations. For about 10 interventions, 4 to 5 hours every day for a month. With our system, we run 20 interventions in tandem, and we can parallelize that where we can run all of these at the exact same time. It takes about 5 minutes for us to actually, in terms of manpower, to do the assay. We just put them on the robot, and the robot does the work for us. So we could get all of that done in, you know, minutes as opposed to days in terms of hours, and in terms of just the timeline to complete the task, a couple of months versus a couple of years.
And also, I think through the automation also, you minimize risk for mistakes and things like that, because the different experiments are done in the same way. So
and like that assay I was telling you about with this theoretical grad student, all they're doing is just seeing if they're alive or dead or not. They're not looking at movement, behavior, animal-to-animal interactions, animal-to-food interactions. They're not getting a chance to see how the animals are growing through time, or not growing through time, or shrinking. Like they're, like I said, we're up to about 20 phenotypes we measure in parallel for every animal that we look at. And, we've been doing this for about 1.5 years now. We've screened, I think we're over 300,000 animals in that multiple phenotype, high dimensional survival, all the way from birth to death kind of strategy.
Awesome. I guess I'm just super curious, Mitchell, to hear from your side, if you're comfortable sharing, like for Ora Biomedical's strategy, is it around further bringing this cost down another 10x, or is it looking at other organisms and potentially bringing down the cost from, whatever it is now, running it in a manual lab to, 10x lower than it is now?
Yeah, our business model is kind of funny. Like, it's a product of the space and what we know we need to do and what the downstream future is. So one part of what we do is contracted research. That's where the Million Molecule Challenge fits in. Anybody can do research with us. You just do a little micro contract. The other side that we're doing is actually internally discovering and developing these interventions. So we find interventions, we find them in C. elegans, test them in combinations, test them across disease mutants, and then rapidly translate those into as close to human systems as we can. And that's where some of these tissue culture technologies are starting to come online. Ultimately, we see ourselves as the medicine cabinet for longevity biotech. We're going to be the source for the best breakthrough interventions, things that don't just, give a 5 or 10% increase in lifespan, but could give 30, 50, double lifespan, those kind of big, audacious goals, pushing the limits of what a single or combination intervention can do in a physiological system. So I don't know if I totally answered the question, but those are kind of the two arms in terms of the model.
Awesome. No, makes perfect sense. Cool. Jillian, did we have any more questions from the comments that we wanted to get to? Just want to make sure we get to them.
We have maybe more of a scientific one that I can show. So I'll pull it up here. So it says, how does an mTOR inhibitor influence other physiological processes that involve the activation of this pathway?
Yeah, that's a really great question. So yeah, one of the intuitions is that if you're taking mTOR inhibitors, it would have potentially a negative impact on your muscle growth. It's something that's involved in biogenesis and increasing cells. So wouldn't that have a harm in your muscles? It doesn't seem to be the case. There are some nuances in terms of how these different pathways interact in different tissues. So the muscle side of the story, it actually sounds like it might work in our favor. The one where there's still kind of up in the air about is in the immune system. So if you take incredibly high doses of mTOR inhibitors, it has an immunosuppressive kind of outcome. And that's what it was originally approved for. You do these smaller doses and you don't see that broad scale immunosuppression, but it's also not clear that that isn't impacting subtly other aspects of immune function. So you want to be careful. And I think, if people are taking mTOR inhibitors, there's a lot of people doing sort of N of 1 kind of studies right now that capturing that information around like how often they have a cold, how often are they, when they get a blood sample, are they seeing a lot of white blood cells in those samples? Different kind of things like that can help us get at that and educate ourselves. So everything we've seen so far, it seems like it's good. It works on the right side, but there is a good opportunity to be a little, just careful.
And usually one thing that I like to talk about instead of just the mTOR, because mTOR can be a little bit too scientific word for many people. It's more about talking about anabolic processes, which Michelle talked about, that it's about growth and development and reproduction, and the opposite processes of anabolic processes is catabolic processes, which is breakdown and cleanup and things like that. So I think if you push too much to the catabolic side of things, then I think you get the detrimental effect of the catabolism, because that can lead to, for example, that you get too fragile, you get underweight and things like that. So there, I think you can find different measurements to find a good balance between anabolic and catabolic processes. So yeah, for example, looking at the muscle mass and muscle performance can be a good way to see that you're not too catabolic and too anabolic. And the immune system can also be one measurement to look at. And some other intervention things like blood pressure can also be something. If you have a very low blood pressure, that can indicate that you're too catabolic, you're pushing too much the catabolic processes, and the opposite comes to when you have high blood pressure. But you need to take all the measurements in consideration. You cannot only look at one thing to see if you're too anabolic or too catabolic.
Yeah. And Krister, what I find so interesting about what you're saying is also just looking at kind of, I don't know if you want to call it ancient wisdom, but basically looking at how certain religions incorporated this concept into their traditions. So like if you look at Islam, which has Ramadan, and if you look at Judaism, which has Yom Kippur, there were these occasional moments of deep fasting where you did do, strong catabolism and probably activated autophagy, activated the mTOR pathway. But they knew that it was only safe to do this, rarely, right? On occasion, because most of your time should be spent in catabolism. But there were these intermittent periods of anabolism, but there were these intermittent periods of catabolism that actually were beneficial to have in your life, and they incorporated them into the traditions of the religion, which I find so interesting. It's like the natural mTOR inhibitor, basically.
Benji, like this wasn't something I intended to talk about, but that brings it up so well. Yeah, I totally agree with you. And in fact, early life things, early life nutrient stresses actually seem like they have some lasting impacts that can even last into another generation. So if we think about that, we think about the world that my grandparents and maybe great-grandparents of y'all's experienced. They also experienced some of these proximal low nutrient conditions as a function of the world wars and different things like that, or in the US, the Great Depression. So then the question I always think about now is, are we at a high watermark currently for longevity as it stands right now? Are we getting benefits because of that? And if, then it changes the narrative. Longevity biotech and longevity interventions isn't just about improving. It's about maintaining the success that we have. Do you want to live as long as your grandparent? Well, then we better get serious about figuring this out, because they might have been a beneficiary of something that you're not benefiting from.
Yeah, I'm a little bit nervous about what I'm passing on to my children, because it's like, I think about the quality of, I was just talking to someone about this yesterday, like walking around the grocery store, I try and shop just the perimeter, because that's where, all the fruits, vegetables, like actual pure food is. And man, then you think about what's in those aisles and what, we're ingesting. And I'm sure that it certainly isn't paying anyone any dividends. I can tell you that. So Mitchell, super interesting point, and something that I think about as a parent with kiddos, for sure.
Yeah.
The last weird thing I'll say, I promise. So another thing that's really strange about how all that works is there's also a neuronal signaling component. So they've done studies in flies where they're under dietary restrictions, so they should reap a benefit from it. But there are food molecules, they can smell food. That blunts the effect. So like, you're doing good with your diet, you're doing great. You drive past KFC and you smell the delicious chicken smell. Have you undermined your progress because your neurons know, hey, there's food around, stuff's great, you should go eat.
Wild.
Stop it.
Wild.
That is intense.
Can't even smell the cheeseburger.
I was gonna say the movie theater popcorn is like stressing me out.
It's like secondhand smoke. Oh my God, that's terrible.
Yeah.
Oh no.
Anyway, we've kept you guys over time. Krister, I know it's getting pretty late your side. We're in between you and the weekend, but yeah, no, I really just wanted to show appreciation for you guys taking the time to speak with us. We'll have a lot of very exciting announcements coming soon that kind of blend together all the different topics that we've talked about, but we'll keep it under wraps for now. Hope that this conversation brought a lot of awareness to how important the work that these two are doing is, and more to come. But yeah, Jillian, anything else to close us off with?
No, I just want to say thank you for your time and honestly the work that you guys are doing, because it's honestly to me like I don't want to say it's probably not supply and demand, but it's like, Mitchell, without the work that you're doing to build the tech, and Krister, without the work that you're doing to find all of these different things that you want to test and meeting in the middle, this can't accelerate. So just hats off to both of you. Thank you to both of you for leading the charge. And I also want to thank you too for taking the time to answer community questions. There's 4 of us on this screen, but there's a ton of people outside these walls that are really passionate about this topic. And so bringing the community in, having these conversations is how we grow awareness for what you guys are doing and invite more people into it. So thanks for the time at the end to work in a few of those.
Big thanks.
All right, we'll be talking soon. But yeah, thanks everyone for listening.
Take care.
Okay,
Bye-bye.